Hello and welcome to the webinar today on Lost in Translation, a story of immigration, mechanisms of tau dementias and other demons, brought to you by the Early Career Research Award, supported by the journal Future Science OA. My name is Francesca and I will be your host for today's event. I'm the head of Open Access for Future Science Group, publisher of the Future Medicine and Future Science collection of journals, and the managing editor of the biomedical open access journal Future Science OA. 2017 saw the launch of the Early Career Research Award, which recognizes outstanding early career research in biomedicine. We are very pleased to announce Joe Abisambra from the University of Kentucky as the well-deserved winner of the award. He was chosen from over 30 excellent nominees by a combination of our expert judging panel and over 1,000 voters. He will be presenting today on his career journey thus far and his exciting research. Joe is currently an assistant professor at the Sanders Brown Center on Aging at the University of Kentucky, USA. He began his scientific career researching Alzheimer's disease at the University of South Florida in the early 2000s. His training focused on Alzheimer's disease and related dementias, and he began his independent research program at the University of Kentucky in 2013. Since 2010, he has contributed close to 30 manuscripts, mentored more than 35 trainees and junior faculty, and received awards from federal, private, and non-profit sources. Joe and his team have narrowed the breach in understanding how protein misfolding causes memory impairment, and in doing so, the Abisambra Lab's work has already identified several therapeutic targets for dementias. Thank you again for joining us today, Joe, and once again, congratulations on winning the award. I'll now hand over to you for your presentation.
Thank you so much for the opportunity to give this webinar to present our work and highlight our passion and interest in neuroscience research. It is truly an honor to have been the recipient of this award, and I'm very excited to share our work with you. The main objective of our laboratory is to establish the molecular mechanisms leading to dementias. These dementias have been narrowed down in our work to tauopathies, and these diseases, which are 25 known neurodegenerative disorders, sharing the same pathological hallmark, which is the abnormal aggregation of the protein tau inside brain cells, has really fascinated our group because of that common pathological mechanism. The other major component of our research program is really founded on this curiosity for how the brain works. And I'll start talking about that from my story as it began in my family. My scientific career began as a child. I grew up in an environment that allowed me to be curious and ask questions. My parents fostered this kind of environment where I wasn't shy, I wasn't allowed to be shy, I was really inspired to ask these questions about nature and the environment around us, how things worked. And this inspiration solidified with both of my grandparents. On my father's side, my grandfather was a chemical engineer, and he fostered that curiosity to understand the environment, the world, the physics, and the chemistry around us and how that impacted our lives. On the other hand, on my mother's side, my grandfather, being a psychiatrist, taught me a lot about how the brain works. But even more fascinating to me was to identify how much of the brain we don't know. And that's where I became really, really curious about science. In fact, my grandparents gave me my first microscope, and I remember as a kid going out collecting leaves and dirt and looking at it in my microscope. So those are fond memories I have that have led me to my career. I'm originally from Colombia, where I spent my formative years in 3 different cities: in Santa Marta on the Caribbean coast, in Buenaventura on the Pacific coast, and finally in Bogota. Colombia is a very varied country with different cultures and idiosyncrasies. And I believe this marked me as an individual and as a scientist. Today, I have the opportunity to interact with people from different cultures, different idiosyncrasies. And even more surprising is to handle the flexibility of science, hypotheses, and questions, and different ways to interpret the data. Academically, my trajectory in science began at age 20 when I moved from Colombia to the United States to pursue a career in science. Research is a luxury of developed countries, and I decided at age 20 to move from Bogota to Florida in order to obtain a Bachelor's of Science degree in biology at the University of St. Leo. And then I obtained a master's degree focusing on biochemistry and molecular biology from the University of South Florida. In 2010, I received my PhD degree from the University of South Florida. For 6 years, I trained in learning about Alzheimer's disease, and I focused on studying the impact of the low-density lipoprotein receptor on Alzheimer's disease progression. My work was done in the laboratory of Dr. Huntington Powder, who at the time was director of an Alzheimer's disease research center at that university. Then I stayed in Tampa doing a postdoc with the late Chad Dickey, where I focused on tau pathology. And this really was inspired by the fact that amyloid beta, the peptide responsible for plaques in Alzheimer's disease, and tau, which is the basis of tau tangles in Alzheimer's disease brains, shared a common hallmark, which is that these peptides and proteins adopt abnormal conformations. In the spring of 2013, I moved to the University of Kentucky, where I founded our lab, and we've developed a strong research program. Today, we have 8 people in our lab. Our group is composed of a senior scientist, 2 graduate students, 3 amazing technicians, and 2 undergraduate volunteers. But my continued inspiration is my family. This is the most recent picture that I got from my wife, Chantal, with our 3 kids, Gabriela, Miguel, and Alejandro, sharing and wearing our Colombian pride jerseys once we qualified for the World Cup. Okay, let's get back to science. So, as I mentioned earlier, the overall objective of our research program is to establish the molecular mechanisms leading to dementias. And I've divided the continuation of this presentation into 2 parts. The first one is an example of how we're using our resources and tools to identify therapeutic targets for tauopathies. And in the second part, I am going to demonstrate some of the work that we've done developing new technology and adapting new tools to existing technology in order to develop both a powerful and sensitive diagnostic tool as well as giving us information about how the brain works. What is dementia? It's defined strictly as a decline in mentation, but more commonly it can be explained as a reduction in mental activity, defined as specifically an impairment of cognition that interferes with daily tasks. This encompasses a loss of coherent speech, object identification, motor tasks, and that can be seen in patients suffering from Parkinson's disease, and an ability to form abstract ideas. The first patient described to have dementia or be demented was King Lear, described very detailed in Shakespeare's work, despite being a fictitious character. So when I'm outside of the lab, people ask me, what do I do? And I say that I do Alzheimer's disease research. They immediately become interested and say, hey, doc, I think I have Alzheimer's disease because I can't find my keys. I forget people's names, phone numbers. All of these events are normal. I try to explain that there's a normal decline in memory as we age. And this is part of it because we lose brain plasticity. And our brains are actually shrinking a little bit. We're not as good as we used to be learning and remembering. But there is pathological loss of cognition, and that's evidenced here by the self-portraits by an artist called Willem Utermohlen. In 1967, in the top left corner, you can see a self-portrait done very detailed and with a very high-quality perspective. In 1995, Willem Utermohlen was diagnosed with Alzheimer's disease, and he vowed to draw a self-portrait every year thereafter. And you can see, as time went by, the use of color, the details in his portraits, and some of that self-perception had declined to see some devastating impact of the disease on Willem's brain in the year 2000. Alzheimer's disease is just one of 25 known neurodegenerative disorders that share the common pathological hallmark of having abnormally folded and aggregated tau inclusions inside brain cells. On the left-hand side, I'm showing you an immunofluorescent stain of an Alzheimer's disease brain with an antibody that detects pathological tau. And these flame-like structures appear inside the brain cells of individuals with Alzheimer's disease, frontotemporal dementia, corticobasal degeneration, and you can go down to the list seeing chronic traumatic encephalopathy, traumatic brain injury, etc. There is an urgent and critical need to understand the molecular mechanisms leading to tauopathies. These diseases are the largest cognitive threat to our aging population. Aging appears to be the #1 risk factor for these diseases. There is no cure for tauopathies, and current treatments stave off symptoms only for a brief period of time. On average, patients report having improved quality of life because of the treatments for only 6 months. After that, the treatments are largely ineffective. Alzheimer's disease is the most common tauopathy. If everybody listening to this webinar reaches age 85, half of us will have Alzheimer's disease. That has social implications because the other half will likely be caring for someone with Alzheimer's disease. Economically, this disease will have devastating consequences. In the United States, it's expected that by 2050, we will be spending $1.2 trillion per year simply to preserve the patient's quality of life, which is constantly deteriorating. $1.2 trillion represents 1/3 of the annual federal budget in the United States. And the more concerning component here is that every clinical trial has failed. Therefore, it is crucial to understand the molecular mechanisms of disease and of these diseases in order to identify novel therapeutic targets. Alzheimer's disease was described over 100 years ago for the first time by Alois Alzheimer. He was a psychiatrist and neuropathologist, which gave him the unique opportunity to meet and treat his patients. In this case, he met Auguste Deter in 1902, treated her for 4 years, and once she passed away in 1906, he had, as a pathologist, access to her brain. He stained for different proteins, and here we're showing 2 of the major hallmarks that confirm the diagnosis of Alzheimer's disease still today. The extracellular plaques are composed of amyloid beta protein, A-beta, and they give a toxic environment outside of the cells. Meanwhile, tau tangles form aggregates inside of the cells and essentially choke that neuron from functioning properly. Our work, because of that need to identify new therapeutic targets, we have focused on intracellular mechanisms by which pathological tau induces cellular dysfunction. And we have focused on a central organelle for cellular function called the endoplasmic reticulum. We're very interested in the ER because it contacts many organelles around the cell, and it seems to be a central point of many functions that are necessary for neurons to allow us to form new memories and recall those memories. One of our major contributions to the field was the discovery that tau associates with ER proteins. And in doing so, it affects the function of these ER proteins. We identified that tau blocks endoplasmic reticulum-associated degradation, or ERAD, preventing the destruction of misfolded proteins. As a result, these misfolded proteins accumulate outside of the ER and prevent it from performing all of its other functions, such as participating in calcium homeostasis, trafficking proteins to the Golgi apparatus or the plasma membrane, interacting with mitochondria, and lipid and carbohydrate metabolism. Ultimately, what we see in these cells is the chronic, sustained activation of the unfolded protein response. And one of its jobs is to slow down translation. However, neurons are quite susceptible to the inability to make new proteins, and therefore their synapses will not work. Tau is a challenging protein to work with. Here I have a schematic showing that tau, as a linear structure, undergoes many translational and post-translational modifications. For example, the 4 regions on the right side of the protein are microtubule binding domains. And the darker green fraction is a splice variant that can be reduced in the full size of the protein, preventing it from associating with microtubules with more affinity. Also, all the red spots represent all the phosphorylation sites on tau that have been studied very carefully. However, other post-translational modifications, such as acetylation, ubiquitination, nitration, methylation, glycosylation, and even cleavage have not been studied to the extent of phosphorylation. That said, there is an emerging concept that tau adopts other functions aside from stabilizing microtubules, and those other functions are regulated by these post-translational modifications. So one of our first studies while we were at the University of Kentucky was based on identifying changes on ER proteins as a consequence of their interaction with tau. One of the biggest strengths of the university is an outstanding world-class brain bank. We have over 500 samples, and we have shipped over 2,000 of these brain samples accompanied with blood and CSF from patients in our community. I, as a researcher at the Sanders Brown Center on Aging, have unique access to these tissues, and I was able to begin our studies by staining for the proteins in this slide. I'm going to show you in the top 4 panels an immunofluorescence staining for total tau in green, calnexin, which is an endoplasmic reticulum protein, in red, and a merged image in panel G. You can see the yellow overlapping signals showing that tau and calnexin overlap, demonstrating that there's some association between the 2 proteins. In panel H, I'm showing the mathematical rendition of the colocalization of these 2 proteins. When we can extrapolate the pathological tau component in panel I by staining with PHF1, we see how that also interacts with calnexin. again stained in red in panel G, and in the overlapping signal, which turns yellow in panel K, demonstrates that these two proteins overlap. We further corroborated these results with biochemical approaches. Here we wanted to enrich the proteins from the endoplasmic reticulum in a sample so that we can identify whether tau was in that preparation or not. To do that experiment, we performed subcellular fractionations from human brains, again, Alzheimer's disease brains, or non-demented age-matched controls. We isolated this microsomal fraction. And from here, we isolated tau to identify all the other proteins that formed a complex with tau on the endoplasmic reticulum. A description of that experiment is presented in this slide, where, again, we used human control in Alzheimer's brains, extracted all the endoplasmic reticulum, and then performed co-IPs or co-immunoprecipitation assays, where we isolated tau or actin in each one of these samples. We were focused on the tau complexes, which would pull down not only tau, but all the endoplasmic reticulum proteins that bound to tau. And we use actin as a negative control. Then we ran these samples on the gel that you can see in blue, and every single one of these boxes highlights a band that we then cut out, lysed, and analyzed using a proteomics approach with liquid chromatography and tandem mass spectrometry. We did this experiment with two different antibodies that identified different regions on tau to corroborate these results. We identified over 216 proteins that associated with tau. And what was really surprising was that there were many more of these proteins that associated with tau in the Alzheimer's disease brains. Surprisingly, only 48 of them were common between the control and the AD brains, or Alzheimer's brains. But again, a major interest of our work is to identify changes in the pathways and the mechanisms. So we speculated that if tau associated with these ER proteins, then those functions that those ER proteins would be dedicated to would be affected. And here I'm showing on the left a pie chart of all the tau-associated proteins and the functions that they perform. We were very interested in the largest slice in blue of the proteins that participate in RNA translation. And we further evaluated their affinity for tau in Alzheimer's and in control brains. And as can be evidenced on the chart on the right, the vast majority of those RNA-binding proteins associated with tau in the Alzheimer's brain and not in the RNA-binding proteins of the control. This discovery was fascinating because we had already identified, and other groups, the importance of protein synthesis for the formation of new memories and the recall of those memories. And while the association of tau with ribosomes was first described 30 years ago from the Skip Binder's lab in 1987, and further confirmed with other publications over the years, the consequences of the tau-ribosome interaction were largely unknown. So we postulated a very simple, straightforward hypothesis, and that is that a ribosome, on the left, would normally translate RNA into new protein. But in the presence of pathological tau species, such as these red and blue ribbons, there would be a high affinity for ribosomes, and therefore translation would be impaired. To test this hypothesis, we went back to our biochemical approach, where we performed subcellular fractionation of human Alzheimer's disease and control brains. And now we're focused on two specific fractions, one that does not contain ribosomes, and another that only contains ribosomes, both shown at the bottom of the slide. And we compared them to a whole cell lysate at the top. we performed Western blots using the fractions of the human brains. And here I'm showing in panel A the first 3 lanes with samples from a control brain, and the last 3 lanes showing samples from the Alzheimer's brain. Lanes 1 and 4 correlate because they have the samples from the whole cell lysate. Lanes 2 and 5 correspond to the fraction that do not have ribosomes. And lanes 3 and 6 correspond to the fraction that only contains ribosomes. The bands represent the amount of tau that's present in these fractions. And the most dramatic difference is present in the shift of the presence of tau from the fraction in controls of the only ribosomes, that is to say lane 3, and the only ribosomal fraction in the Alzheimer's brain in lane 6. That smear at the top of that gel corresponds to pathological tau, as evidenced in panel B. That smear is the conformation of paired helical filaments that are distinct structures of Alzheimer's disease brains. At the bottom of panel B, we're showing Western blots for P0 and Del28, which are both ribosomal proteins, and they only are present in the ribosomal fractions. Meanwhile, HSP70 is a control for cytoplasmic proteins, and they are present in the brain lysate and no ribosomal fractions. We further corroborated these results and added the other toxic conformation of tau corresponding to tau oligomers. These are now known as the more toxic tau conformations, and they're depicted here in red. When we co-stain for tau oligomers with an immunofluorescence stain for a ribosomal protein S6, shown here in green, we also see overlap in panel E, represented by the yellow signal. So these data also demonstrate that tau oligomers associate with ribosomes. But again, for 30 years we've known that these interactions occur, and they occur in disease, but we didn't know why. Our hypothesis was that tau associates with ribosomes and prevents their function of translating RNA into new proteins. So to test that, we used 3 in vitro assays that I'll present to you in the upcoming slides. The first one consists of a cell-free assay, where we had 96-well plates, and in each one of those wells we added the machinery necessary for translation to occur. That is, ribosomes, initiation factors, elongation factors, ATP, etc. In every well, we added a plasmid, DNA coding for GFP, green fluorescent protein. And then we're able to put these plates for 6 hours at 30 degrees and see the rate of translation of production of GFP in real time. Now, the variable in each one of these wells was the addition of toxic oligomeric tau, or control proteins. In panel A, I'm showing the curves of GFP signal that was detected over 6 hours. And in panel A, we're showing in red the impact of tau oligomers on the production of GFP signal. And that is reduced by over 30% compared to the control, which is bovine serum albumin, or BSA, in the purple curve. We then wanted to determine whether this effect was unique to tau oligomers or other oligomeric proteins. So in panel B, we performed a similar experiment, only instead of using tau oligomers, we used alpha-synuclein, which is a protein involved in the pathology of Parkinson's disease, or insulin oligomers. And we found that there was no change in the rate of production of GFP compared to BSA control. These data suggest that tau oligomers can specifically interact with ribosomes and impair their ability to make new proteins. We then tested our hypothesis in an immortalized cell culture model of tauopathy. We developed these cells in Chad Dickey's lab, and I call them the inducible HEC cells. They are HEK, human embryonic kidney cells. They're immortalized, and they stably express tau. The added advantage of these cells is that we can induce tau expression as soon as we add tetracycline. So in this figure I'm showing how at time point 0 there is no tau present. But as soon as we add tetracycline, tau levels increase for up to 96 hours. Using these cells formed a technique called surface sensing of translation, or SUNSET. This is a non-radioactive pulse assay, and it's based on the use of pyrimycin, an antibiotic. It turns out that pyrimycin is structurally similar to tRNA. So when we add it to the cells, it fools the ribosome into thinking that it is tRNA, and it jams the ribosome and sticks to every single new protein that's being made at that point in time. So our experiment consisted of inducing tau expression, adding pyrimycin, and then harvesting the cells. We induced tau expression for different time points before we added pyrimycin. And in panel C, I'm showing expression of tau for 24 hours, 48 hours, and then 72 hours. Total levels of tau and PHF1 increased over time as expected. But pyrimycin, which represents the abundance of new proteins being made, was reduced. That is to say that as pathological tau increases, the synthesis of new proteins decrease over time. But then we decided to do a rescue experiment to confirm that these effects were specific to tau production. So in panel D, I'm showing how we induced tau expression for 24 hours or 96 hours in the ON system in lanes 1 and 2. And you can see that pyrimycin decreased over 4 days as expected. And that it corresponds to that increase in PHF1 pathological tau. But then we removed tetracycline and therefore reduced the ability of these cells to make tau. And we see that pyrimycin levels were restored as pathological tau was eliminated. Our third in vitro model corresponded to the generation of primary neurons from a tau transgenic mouse model. These mice were given to us by Dr. Jada Lewis, who astutely added an inducible feature to these mice as well. It's a TET-off system. But for the purposes of these experiments, I will mention that these mice express a human mutant form of tau, which is P301L, and that is associated with frontotemporal dementia. In the panels I'm showing in the dark stain how the transgenic brains have very dark affiliation or association with tau tangles. Meanwhile, the non-transgenics do not have the presence of these tangles. And in the western blot, I'm showing that human tau is overexpressed significantly in the transgenic brains. In this experiment, we plated primary neurons, and in panel G, I'm showing 3 wells using proteins from non-transgenic primary neurons compared to transgenic primary neurons. We performed our pyrimycin SUNSET assay, and it's evident that the non-transgenic cells had more pyrimycin than the transgenic cells, suggesting that, again, the cells that have pathological tau have a reduction in protein synthesis. Interestingly, and linking this phenomenon to function, we measured the amount of PSD95, which is a protein that participates in synaptic function. And we saw that the transgenic cells had a stark reduction in PSD95. These data are quantified in panel H, showing how pyrimycin and PSD95 are reduced compared to their controls in the transgenic cells. Now, what was really interesting is we performed RT-PCR experiments to quantify the amount of RNA that's being made for PSD95, and that's in panel I. And we can see that the amount of PSD95 RNA is increased by about 2.5-fold in the transgenics. So RNA is increased and proteins are decreased, suggesting that translation is stuck. So to summarize this component of the presentation, I'm showing how under normal conditions, tau associates with microtubules and stabilizes them. Ribosomes come along and they're able to perform their normal function of translating mRNA into new protein, and that contributes to normal memory. Meanwhile, under conditions that are not entirely clear yet, pathological tau forms by the abnormal dissociation from microtubules, and these pathological tau conformers then associate with ribosomes and prevent them from making new proteins. Our results are summarized here. I've shown you data indicating that tau associates with ER proteins, and the most common of these proteins participate in the regulation of RNA translation. I've also shown that pathological tau interferes with ribosomal function using 3 in vitro assays: a cell-free assay, an immortalized cell assay, and a primary neuron assay. These phenomena occur in Alzheimer's disease, and they're related to memory loss. This is a molecular mechanism linking tau and memory loss, and it could be an early event in tauopathies. We are currently performing studies to corroborate these results using in vivo models. One of the major challenges in the field is the identification of biomarkers. Part of the reason why clinical trials have failed seems to be because identification of patient populations suffering from the disease is occurring too late. These brains are already affected by Alzheimer's and other dementias, and as a result, the clinical interventions are not effective. Our lab is developing and adapting new technologies in order to improve on the identification of biomarkers for these diseases. Some of the challenges that are presented today involve the inability to identify early biomarkers, and therefore we are unable to perform early interventions. Current biomarker assays require the invasive procedures such as extracting CSF using lumbar punctures. While there is tremendous promise in the development of PET tracers for plaques and tangles, these are still based on radioactivity and dangerous for the most part with repeat imaging. Also, PET imaging is costly and there is limited access. We have focused on adapting manganese-enhanced MRI using already FDA-approved manganese compounds, which gives us promise that we can move forward effectively. So we've proposed to use a sensitive, inexpensive, and quantitative approach that would be non-invasive, allowing us to perform longitudinal studies, perhaps in patients every year, every 5 years, and in doing so identify changes in neuronal function over time. Another major advantage of being at the University of Kentucky is that we count on tremendous resources such as the MRISC, which is the Magnetic Resonance Imaging and Spectroscopy Core. And we've adapted manganese-enhanced MRI for these techniques. The reason we use manganese is because divalent manganese enters voltage-gated calcium channels, simply because it's similar in size and identical in charge to calcium. Calcium influx into the cytoplasm is necessary for neurotransmitter release. So, manganese enters cells through calcium channels, and they support exocytosis of neurotransmitters. Therefore, it supports neuronal function, broad neuronal function. Now, a major advantage is that manganese is paramagnetic, so we can detect manganese in cells. The way the technique works is presented here. We have these heat maps representing the relaxation time in T1, and the reciprocal is the R1 value. You can see in yellow the absence of manganese, which gives us our baseline in panel A. In panel B, I'm showing you the image of a brain of a mouse 6 hours after injecting manganese chloride in the mice. And you can see now that the areas in red represent those regions where manganese has been taken up in the brain. Because of the high resolution of the equipment that we have here, we can identify small regions of the brain and even subregions of the hippocampus to perform quantitative analysis. Again, we can perform longitudinal studies. Here I'm showing the shape and the distribution of manganese throughout mouse brains at 2, 3, 6, and 10 months. And we can see how the shape of the brain is changing over time, and the distribution of manganese also changes over time. So for these studies, we've used our tau transgenic mice that I presented earlier. These mice overexpress human P301L disease-associated mutant tau. In the course of the disease, we see tau expression at around 2.5 months, progressive tau pathology and cognitive impairment ensuing after the development of tangles, and then deficits in neuronal plasticity. By 5.5 months, we see overt neuronal loss and significant atrophy. Here we can see a normal brain in panel A, presenting a very plump hippocampus, and this area that's highlighted in the rectangle corresponds to the dentate gyrus of the hippocampus. In panel B, I'm showing you a 10-month-old tau transgenic mouse. The parenchyma in this brain, all the tissue, is shrinking quite significantly compared to panel A, which is the age-matched control. We can perform our R1 values and measurements, and you can see that everything in blue in panel B corresponds to CSF, filling in that space of the shrinking brain. This figure shows how at early time points there is tau pathology developing in the different brain regions, such as the CA1 in panels A and D, CA3, panels B and F, the dentate gyrus in panels C and G, and the cortex, superior medial cortex in panels D and H. All the dark staining corresponds to tau pathology, which is very light in panels A through D, but it certainly becomes more identifiable in panels E through H. We've quantified those results in panel I, showing that there are significant differences in the accumulation of tau pathology at 3 months compared to 2 months. And in panels J, K, and L, I'm showing a close-up of these tauopathic inclusions. Now that I've shown pre-tangle pathology at early time points, 2 and 3 months, I can also show how our manganese-enhanced MRI can detect changes in broad neuronal function as a consequence of the development of this early tangle pathology. Here I'm showing how the 3-month brains look slightly different in the controls, but they are very different in the transgenics, especially when we compare at 3 months the control to the transgenic. At 2 months, when there's very little or no tangle pathology, we don't see that the brains, panels A and C, look that different. The panels B and D, at 3 months, where more tau pathology is present, we see more changes in the manganese signature. And that can be evidenced in the hippocampus, that area that's highlighted in the rectangle, where much of the hippocampus in the transgenic is red, while the 3-month in panel B is very plump, yellow and blue. This is an example of the application of manganese-enhanced MRI. Panels A and B correspond to normal brains. Panels C and D correspond to transgenic brains. And these brains have been treated with either a vehicle control or a tangle-reducing compound called 414. Panels A and B show that the drug didn't really change the distribution of manganese. However, panel C shows that the brain is very much reduced in size, and especially in the hippocampus. You already see some atrophy demonstrated by that blue point in the middle of the brain. This is a transgenic mouse brain, a sick mouse, that has been treated with a vehicle control. But when we treat the mouse with the 414 tau-reducing drug, we see that the hippocampus and the brain tissue looks much more similar to the normal brains in panels A and B. As I mentioned, a powerful feature of this technology is that we can quantify the manganese signal in the brains. And I'm showing those two examples in panels E and F of brain regions of the dentate gyrus and the CA1 respectively. The black and gray bars correspond to the signal of R1, delta R1, in the non-transgenic control brains, whether treated with the vehicle or the drug. The red column represents the transgenic brain that was treated with vehicle, and you can see that there is a significant decrease in the signal in delta R1. But that signal is rescued upon treatment with the tau-reducing compound. This figure demonstrates the impact and the power of our manganese-enhanced MRI technique and parametric mapping for quantification. And it can tell us how brains are accepting and improving based on the treatments that we can give to, at this case, the mice, and hopefully in the future to patients. Another example is performing manganese-enhanced MRI in the brains of mice that have suffered a traumatic brain injury or a head injury. In panel D, I'm showing a pre-injury scan. In panel E, I'm showing an injured brain after 7 days treated with a vehicle. And then in panel F, I'm showing a brain image with a T1 of a mouse that had been injured and treated for 7 days with the tau-reducing compound 414. We are currently performing studies to understand what the effect of this drug is in head injury. But what's quite surprising is that in the MRI, we can see that there's a change in the morphology of that brain. For example, the arrowhead is pointing in panel E to blue space, perhaps corresponding to inflammation. And that inflammation seems to be reduced by the treatment of the drug, as depicted by the green arrow in panel F. So to this part of the presentation, we've adapted a technology called manganese-enhanced MRI with parametric mapping to measure changes in broad neuronal function. This is a sensitive, reliable, and inexpensive technique that is well suited for longitudinal studies. And we are currently dedicating resources and efforts to make this approach translational into the clinic. So I will end this presentation by acknowledging all of the collaborators and our funding sources that have allowed this work to take place. Again, these are just a few examples of the work that we're currently performing in the lab, but there's so much more that we're engaging into with different diseases and different technologies. These efforts are supported by Peter Nelson, Heining Zhu, Muriel Vansberger, Kathy Satman, Brett Smith, Fred Schmidt, John Gensel, and Dave Powell with the Neuropathology Core, MRISC, and Traumatic Brain Injury Studies. Much of this work was inspired by my former mentor, the late Chad Dickey, Jada Lewis, who gave us the mice, Peter Davies, who contributed the PHF1 antibodies, and Rakesh Kayed, who contributed the T22 tau oligomer antibody. And of course, our funding sources from the NIH, the Department of Defense, GlaxoSmithKline, and the Alzheimer's Association. Of all of our collaborators, we have our lab. These are really the curious and fantastic technicians who are dedicated to moving our studies forward, from the technicians to the students to the career scientists. They have really been the tremendous motor moving this work forward.
Thank you for that excellent presentation, Joe. I hope our audience found it as interesting as I did. If you have any questions for our presenter or would like to learn more about the Early Career Research Award or its supporting journal Future Science OA, please follow the link shown to our website or contact me at f.slake@future-science.com. I would like to once again thank Joe for his talk and to you, our listeners, for your time. Remember, we will shortly send you a link to the recorded version of the talk. Thank you for your time today and goodbye.